Hello everyone, this is your Prelab discussion for Exercise 8, Tree Thinking Concepts Using Miskin. As a culmination to your evolutionary biology laboratory, you will be recreating the evolutionary and phylogenetic trees of select plant and animal studies. Remember, phylogenetic trees depict the inferred evolutionary relationships among a set of taxa. It shows lineages, highlights the monophyly or the non-monophyly of the investigated taxa, and infers identity of questionable organism. Lamarck calls it the ladder of life, with the assumption that those at the bottom are of ancestral, less adaptive traits, while those at the top or on the tips are of a more derived, fit characters. Darwin refers to phylogenetic trees as the tree of life, the one which unites every single entity into the universal idea of evolution. Phylogenetic trees contain these basic parts: the root, the hypothetical start of the investigated relationships; the node, the bifurcation which indicates a speciation event; and the terminals or taxa, hereby shown at the tips of each branch. Trees can be presented in many shapes and forms depending on the goal of the researcher. It can be the usual bracketed with either an increasing or decreasing arrangement, or with branches treated as with equal or unequal lengths. A rooted circular view, or even an unrooted view, is used in many studies. Trees can also be differentiated into its main sources of phylogenetic data. Combined and/or separate analysis of morphological and molecular data can be used to infer relationships among taxa. The use of each dataset must be supported and must also provide sound inferences for it to be accepted by the scientific community to which it is presented to. These trees, with added geologic, fossil, and occurrence data, can be used to recreate phylogeographic relationships. Such analysis is of great use in concluding the identities of species with wide dispersal ranges, finding the clues as to how it was able to colonize a certain area and how diverse their identities are from each other. Divergence times also gives clues as to how groups of organisms evolved, developed, and diverged over a longer period of time, sometimes ranging from millions to eons of years. With the goal of tracing relationships and evolutionary history, phylogenetic trees can be categorized as either a species tree or a gene tree. A species tree presents the relationship among species or higher taxa, with each bifurcation presenting a speciation event. A gene tree, on the other hand, presents relationships among different copies of a gene. First, paralogous genes, or the genes copied from individuals of the same species, can be mapped on a tree wherein each branch would show duplication events. A gene can be considered a species tree if it presents orthologous genes, or the single copy genes sampled from two or more species. Since there are multiple species investigated, each major branch is a speciation event. Lastly, multiple copies of paralogous and orthologous genes can also be presented in a single tree. Shown here are both species and gene trees of Galaxia's species. Observe how each species is differentiated, speciation events, in the species tree, while the use of paralogous and orthologous genes from the said genus cemented the identities of the samples. As previously mentioned, a tree, no matter what data it used, and regardless of what level of identification it is trying to interpret, it must be well supported and must be widely accepted in a variety of analyses used for phylogenetic inferences. Here presented are the maximum parsimony, maximum likelihood, and Bayesian inferences of the same set of data. These trees will be the goal for the said activity. As mentioned, you'll be utilizing Miskit. This is an extendable, open-source, modular software used in studying evolutionary biology. Comparative data for phylogenetic analysis, population genetics, and even non-phylogenetic multivariate analysis can be conducted using this single software. Most especially, character trace evolution of both morphological and molecular data can be studied in this software too. Your main goal is to conduct the analysis presented in your laboratory manual. In the end, a more in-depth understanding of phylogenetic relationship and as well as a thorough review of your tree thinking concepts will be tested in this exercise. You have to follow all of the instructions presented in your manual and you will be good to go for this exercise. Before you leave, kindly note that we have prepared a recap activity to see whether you still remember how to read and interpret your phylogenetic trees of different levels. Answer the following questions using the discussion board your instructors will be posting together with this pre-lab discussion. All answers will be given by the end of the week. We bid you all good luck and have a great time with your exercise. Stay safe and thank you.